a sec. Okay. Thank you very much, Han, for agreeing to give Build a Cell seminar, and you can get started.
Okay, thank you, Kate, for inviting me to this seminar series. When I first got her invitation, I was kind of hesitated because my research topics are not for this build-to-cell synthetic community, but I accepted her invitation because somehow my work may give you some ideas and future directions when you engineer synthetic cells or cell-free systems. I'm a device engineer making miniaturized sensors, electronics, and batteries by using microfluidic techniques and microfabrications. Especially, I'm using living bacteria as a biocatalyst for powering, sensing, and synthesizing. The bacteria I'm using have a special capability of transferring the metabolically produced electrons to external electrode. We call this exoelectrogens. And actually, this phenomena was discovered 100 years ago. And the people think that using this bacteria can solve our energy and environmental crisis we are facing right now. My field is a little bit different from what people are working on in this field. While most of them are working for large scale applications as a solution for energy environmental crisis, I'm developing miniaturized systems with this special bacteria for some unique applications. But before I start my research presentation, let me briefly introduce myself and my school. I'm from State University of New York at Binghamton. SUNY has 64 campuses throughout New York State. The three largest campuses are Buffalo, Stony Brook, and my university, Binghamton. So we are very close to Cornell University. So SUNY, Binghamton or Binghamton University is actually #1 public college in New York State, and also be known as great public college in the U.S. We have about 20,000 students, and also engineering college having 7 departments is top ranked in upstate New York with a significant financial support from the state and the SUNY. The number of undergraduate and graduate students keep growing. 2 years ago, we got the Nobel Prize winner in chemistry, Dr. Whittingham, who is the distinguished professor in the chemistry department and also affiliated in our engineering college. Because of this, SUNY Binghamton is leading a large northeast center for chemical energy storage, working with many other universities and national labs, and founding the many startup companies in Binghamton area. So based on this SUNY support and other government funding, my research group could focus on developing new bioenergy techniques using bacterial materials. We have created many biobatteries, biofuels, and biophotovoltaics for various applications. Okay, now let me talk about my research. First of all, I will give you general explanation about electrogenic bacteria and what are the current problems with conventional macro scale applications of the bacteria. Then I will introduce new avenues and applications of using these bacteria with microsystems. I will talk about 3 unique applications for powering, sensing, and synthesizing. So as you might know, the electron transfer is one of the most fundamental life processes. Microorganisms like bacteria use this electron transfer for their growth, cell maintenance, and information or energy exchange with their external environment. And some specific bacteria called exoelectrogens can transfer those electrons extracellularly across the cell membrane to external electrode. There are multiple extracellular electron transfer routes through direct transfer, nanowire transfer, or shuttle transfer. There will be many applications of using microbial extracellular electron transfer activities. Harvesting electrons can produce electricity to power other applications. We can treat wastewater or bioremediate the polluted areas by using them as electron donors, or inversely, the electrons can be supplied to those bacteria to reduce carbon dioxide, to produce other value-added gases, chemicals, or biofuels. Bioelectron transfer in a bioelectrochemical system can remove salt from seawater. And because this electricity generation is depending on bacterial metabolism, the output can be used as a transducing signal for biosensing. So even a few years ago, no one doubted that the next generation of renewable energy could come from electrogenic bacteria because of their extraordinary ability to extracellularly transfer electrons from renewable organic materials to external electrode. So with this clear motivation to address the impending energy climate crisis, there will be considerable and continuous research activities on the large scale system development and operation. However, controlling the living bacteria in a macro scale system is very challenging. The output, the power output from the system was way too low to solve our energy crisis, and the output was not reliable, reproducible, and durable. And scaling up the system requires a lot of money and was not manufacturable. And also maintenance and operation was very challenging. As an alternative, the stacking of small units have been proposed, but this one was more complicated for biofabrication, operation, and maintenance. So many leading researchers became very skeptical about this technique for larger scale implementation. But I found the huge potential of electrogenic bacteria when they are with micro scale device platforms. I'm not talking about micro-sized sensors to passively characterize bacterial behaviors. I'm talking about new opportunities of bacteria serving as an integral active machine of microsystems. Their power generation can be sufficient for small low power applications. Their signal can be sensitive enough in microsensors to monitor or detect the presence of some specific molecules. And in a miniaturized engineering platform, they can effectively and reliably produce some nanoparticles, or bacteria itself can be active material for engineered living electrodes. So now I will talk about these three opportunities in the field of powering, sensing, and synthesizing. So first, I will talk about applications for powering. So bacterial electron transfer mechanism can be effectively used to generate power in a bioelectrochemical system. We call this microbial fuel cell, or MFC. So when the bacteria break down the organic food, they generate the electrons and the protons. The electrons are flowing through the external circuit and used for powering the load. At the same time, the protons are traveling through the selective ion exchange membrane and reduced at the cathode with the oxidant. For continuous power generation, the organic food should be constantly supplied. if the organic food is not constantly, continuously supplied, the microwave fuel cell is said to be, biobatteries. So the device will stop generating electricity once the organic food stored in the device is depleted. So basically, the working principle of microwave fuel cells and the biobatteries is the same. The only difference is whether the organic fuel is constantly supplied or not. So normally, potential applications of the microwave fuel cells are based on larger scale, and these are considered as a promising alternative technology that could reduce energy crisis and environmental pollutions we face these days by treating wastewater and remediating the polluted areas. However, it is difficult to see how they can meaningfully contribute to solving either issue in the short term because existing microwave fuel cell demonstrate low performance. Their power is never enough for any potential applications. And also, they experience bottlenecks in scale up. It is too expensive to make it practical. And also, the amount of power generated by the microwave fuel cell is not a linear function of their size. So even if the microwave fuel cells can be fabricated for larger scale applications, their power output and wastewater treatment capacity do not increase proportionally. And finally, the long-term operation of the living bacteria in a microwave fuel cell system requires expensive and energy-intensive fluidic feeding system to replenish bacterial organic food, which will lead to higher energy costs than the microwave fuel cell produces. And also during the operation, many other issues in the fluidic systems, make this technology difficult to be realized. So my idea was really simple. If the biggest issues for microwave fuel cells are low power density, scale-up bottleneck, and maintenance-dependent operation, then a small power and miniaturized microwave fuel cell platforms for short-term operation or a new sustainable platform without fluidic feeding system will be more applicable and potentially realizable. So by miniaturizing the microwave fuel cells with these special features, I could find new and unique applications. So the miniaturized microwave fuel cells can be designed for disposable and the short-term applications so that they work more like as a batteries rather than fuel cells. For example, biobatteries can be built on the disposable paper substrate for powering other disposable biosensors. So in any resource-limited areas like developing countries, you can generate the power from dirty water available in our environment because they include bacteria. Or we can use sweat-eating bacteria living on our skin, or use gut bacteria living in our body to generate the power. Or by using photosynthetic bacteria, you can avoid using the fluidic feeding system because these bacteria can produce their own organic food through photosynthesis. And this miniaturized photosynthetic microwave fuel cell can be self-sustainable and can be most suitable power supply for environmental wireless sensor network for IoT. In this case, I'd like to call these energy technologies as biobatteries rather than microwave fuel cells because the device does not require any organic material replenishment. Of course, we can keep supplying, bacterial organic food, but then, we will have the same issue of maintenance-dependent operation. So that's why I just, focus on, biobattery development without, fluidic feeding system. So let me first talk about, disposable, biobatteries. So my group, for the first time developed a paper, based disposable, biobattery. So our first generation, paper biobattery was developed in 2013. We used a filter paper as a reservoir to contain the bacteria inoculum on top of the carbon cloth electrode. We found out that the paper battery had a very short startup time and instantly generate the power when we introduced the bacterial sample because of the strong capillary force of the paper. As you can see from this SAM image, there were a lot of bacterial cells found in the paper reservoir. And by connecting 4 batteries in series, we could light up the LED, which can be replaced with any low power biosensors. However, our first generation paper biobattery had to use some non-paper layers. So 2 years later, we developed an entirely paper-based biobattery. We developed a paper anode, paper cathode, and paper membrane. And we introduced the origami technique. So, by folding the paper, 4 batteries were connected in series. However, we could not generate enough power for actual applications. The power was smaller than our first generation device. Our next paper biobattery revolutionized the performance. It was Ninja Star shaped device having 8 battery blades. So it can be closed and opened. When it is closed, there is one inlet in the middle so that you can introduce the bacteria. When it is opened, the 8 devices are connected in series so that you can increase power and output voltage. So we could light up the LED for more than 30 minutes. However, as you can see, the device fabrication was really complicated, requiring multiple paper layers. So to solve this issue, we integrated all the battery components into a single sheet of paper. So this is the cross section of the single sheet paper device. As you can see, there's a anode and the cathode and anodic reservoir to include the bacteria and the membrane between the anode and cathode. And the beauty of this technique is that you can just stack them, or you can fold them to provide various combinations of serial or parallel battery connections. And recently we integrated 16 biobatteries on this single sheet of paper. They can be connected through these 2 electrical switches mounted on the paper. And there's one LED in the middle, which can be powered by the biobatteries. So we pre-inoculated bacteria and then freeze dry them before use. So to activate this bacteria, we introduced human saliva and switched it on, and we could light up this LED. The interesting part was that the LED intensity was proportional to the concentration of glucose in your saliva. So this concept can also be used as a self-powered biosensor. So instead of using liquid like a human saliva to rehydrate the freeze-dried bacteria, we integrated media pouch inside the device. So with the finger actuation, the media can be released to rehydrate the freeze-dried bacteria and generate the power. So the device actually powered the LED and the electrical calculator. I don't have time to talk about other platforms of the biobatteries we developed, but let me briefly introduce other biobattery topics too. So other than environmental microorganisms, I was really curious about other human bacteria. I found that many skin bacteria have the electrogenicity, so that we could develop an wearable biobattery. And recently we used Bacillus subtilis as the biocatalyst for the wearable biobattery. As you can see, the B subtilis can transition into a dormant state as spores and can be germinated in nutrient-rich condition. So they can be very innovatively used for on-demand power generation and long-term storage. So with the human sweat, the bacterial spores were germinated, generating enough power. And also we tested many other gut pathogens. Surprisingly, many of them have electrogenicity, so we can develop a ingestible biobattery as well. And also we are developing self-sustainable biobatteries using photosynthetic bacteria, because photosynthetic bacteria can produce their own organic food through photosynthesis. All we need is solar energy. Recently, we developed a biosolar cell that can work independently without any organic fuel introduction, and successfully powered a sensor and remotely transferred the signal to the cell phone. Okay. So, other than power applications, electrogenic bacteria are a promising recognition element for biosensing because they produce quantifiable electricity by sensing and responding to a wide variety of external signals, including nutrients, toxicants, environmental conditions, and many other biological stimuli. Any presence of toxic substances or variations in the environmental conditions can affect the metabolism of the electrogenic bacteria, leading to a sudden change in the output electricity, while a change in specific organic substrate concentration can result in a direct alteration in the output electricity. So these microwave fuel cell can act as a self-powered biosensor for monitoring toxicants, operational conditional changes, or biologically active components. In particular, the miniaturized microwave fuel cells opens broad perspective to radically improve sensitivity, portability, manufacturability, and throughput with a reduction in the response time. So in this section, I will introduce innovative miniaturized microwave fuel cell platforms as a biosensor, which can be most suitable for practical field applications. So the microwave fuel cell based biosensor has attracted increasing attention for water quality monitoring because it can theoretically achieve self-sustaining and real-time water alert system without additional power sources and transducers. However, traditional large-scale application is very limited by several challenges, including long startup time, low sensitivity, low reproducibility, and system instability. On the other hand, miniaturized microwave fuel cell biosensor can be more realistic and practical by improving sensitivity and reliability with excellent control of the microenvironment. So several years ago, we developed a dual channel biosensor integrated detection and reference microbial fuel cell device, providing a reference compensated sensor response so that the signal was stable even with the fluctuation in environment conditions. So by subtracting the response of the reference channel from that of the detection channel, the stable reference compensated response was obtained according to the temperature and the flow rate changes. Actually, this is not possible with macro scale devices. Also, the microwave fuel cell biosensor was very sensitive to one of the toxic materials. So in this case, we selected formaldehyde as a model toxicant. Their current generation decreased sensitivity to various concentrations of formaldehyde. So we first injected 0.003% formaldehyde for 90 minutes, and the current decreases. Then with fresh media introduction, it goes up to the original point. Then we injected 0.015% for 90 minutes, and then current significantly decreased. However, with the fresh media, it does not go back to the original point. And with 0.075%, again, the current does not come back to the original point. Well, actually, the reusability of the microfuel cell as a biosensor is very critical for online and real-time monitoring of water quality. If the microfuel cell cannot be recovered from toxic substances, then the biosensor should be replaced, posing a problem for real-world applications. However, when we injected the toxic substances just for the short period of time, the current was completely recovered. For example, with a very high concentration of formaldehyde, but only for the 10-minute injection, the current came back to the original point after the fresh media washing. This result suggests that our MFC biosensor can be reused under even high level of toxic materials in media if the injection time is carefully regulated. Although the miniaturization of microbial fuel cell can significantly improve reliability, sensitivity, and response time, it is very difficult to allow effective deployments of the microfuel cell in the environment for real-time water quality monitoring. Even the miniaturization cannot avoid many common problems, including biofouling, clogging, and corrosion, which require frequent recalibration of the microfuel cell for stable baseline. And still, microfluidic technique requires considerable power to continuously introduce water to the microfuel cell, hampering self-sustainability and autonomous operation. So in recent years, we developed a disposable, paper-based microfuel cell biosensor as a more practical and realizable application for rapid on-site water quality monitoring. So although the paper platforms cannot provide online and real-time operation for a long-term period, the potential of the device as a disposable, low-cost, and simplified sensor can open up a great perspective to quick on-site monitoring of water quality. So we could eliminate the time-consuming and very expensive pre-treatment procedures of conventional off-site monitoring techniques. So we developed a couple of paper-based microfuel cell biosensors for on-site water quality monitoring, for preservation and long-term storage of the electrogenic bacteria in paper. Pre-loaded bacterial cells were air-dried or freeze-dried, demonstrating long shelf life. So after the use, the paper biosensor was safely and properly disposed of by incineration, potentially removing any risk of bacterial infection or contamination. More importantly, the paper-based microfuel cell platforms could address critical challenges associated with conventional large-scale or microfluidic microfuel cell based biosensors. Because the microfuel cell biosensor can monitor the presence of toxic components in water, the susceptibility of electrogenic bacteria to antibiotics can be innovatively evaluated by monitoring the electricity harvested from the bacterial metabolism. So the microfuel cell based approaches can continuously evaluate antibiotic effectiveness by monitoring extracellular electron transfer and offer early diagnostic analysis by detecting initial and sudden decrease in microbial metabolic activities. So my group, for the first time developed paper-based miniaturized microfuel cell array for rapid, high throughput, and real-time monitoring of antibiotic susceptibility. So the paper-based 8-well sensing array, as the transducer, offered a rapid and simple approach to characterize microbial electric response in the different concentration of antibiotics. So the array generated quantitative actionable minimum inhibitory concentration result within just 5 hours by measuring electricity. And also using micro fuel cell, we also developed self-powered biosensors for diagnostic applications. We created a biosensor using well-known electrogen Schwann onedances for lactate monitoring in human sweat. So because sweat contains high level of lactate, which is the most available carbon source in sweat for bacterial metabolism. And this Schwann species preferably use lactate as their main electron donor. The lactate concentration in sweat can be sensitively and selectively monitored. So if we can improve the specificity of bacteria toward some other biomarkers through genetic engineering, the bacterial self-powered biosensors will be the most powerful device that can dramatically revolutionize public health care. So other than power and sensing applications, bacteria actually are living biofactories that can biosynthesize nanoparticles and produce nanowires during their metabolism. Because of its cost-effective and renewable and eco-friendly features, bacterial biofabrication has attracted extensive attention in the field of nanotechnology. In particular, the electrogenic bacteria are regarded as promising system for green synthesis of metallic nanoparticles during their respiration. And also, engineered living materials can be formed with the electrogenic bacteria for special smart functions. So, first, electrogenic bacteria can transform metallic ions into nanoparticles through their electron transfer, which remain bound to the cell membrane. So for example, palladium and the gold nanoparticles can be produced, and you don't need any toxic or complicated chemical processes to produce nanoparticles. So the palladium nanoparticles were successfully biosynthesized and remained bound to the cell membrane. And actually these bacterial cells with the nanoparticles provided more efficient electron transfer pathways and improved the power generation. So we prepared this miniaturized paper-based microfuel cell and tested their power generation. And as you can see, we could significantly improve the power and current generation. Oh, actually, we could control the size of nanoparticles on the cell membrane by changing the bacterial concentration. With high concentration, nanoparticle size was very small. With the low concentration, the particle size was very big because small number of bacterial cells have to handle the high concentration of metal ions. So after we adjust the concentration of bacteria with the nanoparticles on their cell membrane so that they have the same concentration, we tested their power generation. As you can see, the bacteria with the small size of nanoparticles generated more power than that with the large size nanoparticles. So this is because electron transfer increased in the presence of small size particles, while the large nanoparticles interfere with the electron transfer pathway and the catalytic reactions. So we also successfully biosynthesized gold nanoparticles using cyanobacteria. Actually, we found that gold nanoparticles were working as an effective light absorber to increase photo-excited electrons and as an electron conduit to improve the electron transfer through the cell membrane. So this cyanobacteria biosolar cell, with the intracellular gold nanoparticles, enhance the maximum power density by as much as about 30 times compared with the device without nanoparticles. Even after the long cultivation for more than 120 hours, only a few dead cells appeared in cyanobacteria with the in situ formed gold nanoparticles, which indicate that unsignificant adverse effect on cell viability and providing the feasibility of the modified cyanobacteria as an improved biosolar cell biocatalyst. So finally, the electrogenic bacteria can form a living electrochemical electrode that can enable significant advances in bioelectrochemical systems. So basically, we can create a great performing, very conductive, and very thick layers of electrogenic bacteria that can be mass produced with the new biomanufacturing techniques. And these electrodes will be a fundamental groundwork for all bioelectrochemical applications. And these electrodes will have a controllable functionalities and seamless biotic interfaces. So we successfully created living electrodes by combining controllable 3D printing and the simple electrochemical polymerization technique. So we entrapped and connected the electrogenic bacteria to the conductive polymer electrode, forming a three-dimensional conductive composite material. So the electrode included multiple bacterial species to provide a self-sustainable function. So these co-culture systems provide a cooperative interaction where one species produces a resource that the other species needs, but cannot synthesize. So for example, we formed an engineered living electrode by using three bacterial species for self-sustainable bioelectricity generation. So in this concept, this cyanobacteria on the top layer uses photosynthesis to produce carbon hydrate, which are used as a carbon source by other bacterial communities below. Then these facultative aerobe B subtilis are inserted into the middle layer of this co-culture to produce riboflavin as electron shuttle, which significantly improves the power generation of the exoelectrogen on the bottom layer. Furthermore, the B subtilis can remove oxygen and thus maintain the anaerobic condition for the optimized electron transfer process of these exoelectrogens. So this synthesized co-culture system generated really high power performance for a long period. So this work confirms that biobatteries engineered with a microbial consortium have enormous potential to be sustainable and reliable energy technique. So let me conclude my presentation. The use of electrogenic bacteria had attracted so much attention as a new energy and wastewater treatment technique. However, the larger scale devices are not that practical yet because of many issues. Rather, the miniaturized platforms in flexible, stretchable, or disposable materials are more feasible and realizable at this point. And also, when we consider the number of bacterial cells available in our nature or in human body, using bacterial cells as energy resources for low power biosensors and electronics is very attractive. And bacterial electrogenesis can be potentially used as a biosensor because the generated electric current can be a measure to monitor the presence of specific biomaterials. And the micro-scale biosensing platforms can be much more realizable and practical than the macro-scale counterparts by maximizing the sensitivity with the small background noise and variations in the controllable micro-scale environment. And finally, bacterial electrogenic activities can be precisely controlled in micro and nano systems to synthesize designed nano materials, and their electrogenic capabilities can be engineered to be maximized and self-sustainable in the context of engineered living materials. So currently my lab is supported by 3 NSF and 1 ONR grants and other internal SUNY awards. I also want to thank my students for their hard working, and thank you so much for your attention. Now I can take your questions and comments. Right.
Thank you very much. There is a lot of questions in chat. Can you see chat?
Okay, let me check it. Okay,
If you could read the question. Yeah.
So could the micro cell be used to sense metabolites from cells for biocomputing applications? Yeah. So many other researchers actually use this electrogenic bacteria for biocomputing applications. They actually fabricated some simple circuitry as well. Yeah. And the second question, fascinating talk. Thank you. Could you constitute electron transfer chain of electrogenic bacteria in a cell-free system with? Oh, okay. So this one, I'm not sure. Okay, I'm not the expert in the built cell, or cell-free system fabrication kind of things. Maybe Kate, can you answer this?
I don't think it has been done so far. I think the biggest difficulty would be to reconstitute the membrane proteins. I don't know what membrane proteins make up the electron transfer chain in those particular bacteria, but I imagine expressing and properly inserting those in a membrane would be difficult.
I see.
It would be really powerful if someone did it though, because all those applications that Sean was talking about, we could do that on a non-replicating cell, and that would be fantastic.
Thank you. Okay, moving to the next question. Given photosynthetic efficiencies are much low compared to photovoltaics, do you think we can ever make an efficient electrogenic bacteria-based energy production system? Oh yeah, very good question. Yes, this is true. The conventional semiconductor photovoltaic cells have much higher efficiency than the biological biosolar cell. That's true, but I'm looking for some unique applications for low power IoT biosensors, so that we can sustainably generate power for those IoT applications. Compared to the semiconductor photovoltaic cells, our biosolar cell can continuously generate power even in cloudy days or at night, because bacterial cells can generate electricity even using the respiration without solar energy. So that's the one advantage over the semiconductor photovoltaics. And second advantage is that our biosolar cell is really cheap. Less than $1, while semiconductor and photovoltaic cells are extremely expensive. So my work is not to replace conventional semiconductor industry devices, but to looking for some unique market and unique applications. Okay, next question. Do people look at those biofuel cells for remote applications and space exploration? Yeah. So actually NASA, invested a lot of money in this field, so that they wanted to, exploit astronauts' pees and poops for energy generation in the universe. And also, ONR is actually developing many, microwave fuel cells for some oceanic remote applications. And actually there are some commercially available microwave fuel cells. Do bacteria in sensors or power cells divide? What happens to waste products? Yeah, this is very good question. So that's why when you are deal with large scale applications for long-term operation, well, actually you have to consider this kind of issues. You have to keep controlling the bacterial regeneration and waste products kind of things. But my field for disposable and short-term operation, these kind of problems cannot be a challenging issue. That's why I'm focusing on this special field. Okay, for environmental monitoring, how do you protect against the horizontal gene transfer to wild type organisms, for example, and spreading antibiotic resistance? Yeah, very good question. So one of the advantages of using micro-sized devices is that you can well package the device so that bacterial cells cannot move out from the device, or other environmental bacterial cells cannot attack the bacterial cells for the device. But for the macro scale, yes, this is very significant issue, so we have to consider them. So what type of polymers did you use in your co-culture? So we used PDOT PSS as a conductive polymer. So we polymerize EDOT to conductive PDOT by applying the potential. So have people engineered the combined bacterial cells, like a combined electricity generation with the sensing in one strain, or would this be best done by co-culture of different strains only? Okay, yes, this is true. Many researchers are trying to use engineered and combined bacterial cells for the better performance. But I can say that I'm sorry for you guys working in genetic engineering of bacteria, but whenever I present my work, I introduce genetic engineering as a future technique to address the many issues with the naturally existing bacteria. However, even genetic engineering has many issues too. First of all, the robustness of engineered cells in complex environment show large deviations compared with the laboratory parameters. Furthermore, bacteria can cause unnecessary burdens for the cells and slow down their growth and reproduction. As a result, it's still difficult for engineered bacteria to compete against natural strains in real environment. Concerns of genetically engineered bacteria also lie with their potential threats to our ecosystem and the long-term impact to human beings. Okay, very basic question. Can you connect multiple electrogenic bacteria in series and tap the voltage potential? What's the maximum electric power captured or generated thus far? In the best case, that. Okay, so it depends on the application, of course. We've developed many biobattery stacks with serial or parallel connections to improve the power and current and output voltage. So another advantage of using miniaturized battery is that you can easily stack them or connect them for serial or parallel connections. So basically we could generate more than hundreds microwatt per centimeter square right now, but this one can be improved further. And actually recently by connecting several biosolar cells in an array, we could generate in a milliwatt range for wireless signal transfer. Okay, will microbial fuel cell be able to power medical implants? Yes, so this is actually what I'm working on right now. I'm developing ingestible biobatteries using human gut bacteria. I'm developing a swallowable capsule type biobattery, including human gut bacteria, and it generates about microwatt per centimeter square. So it can be enough power for some low power biosensors implanted. But still, there are many things we have to solve, like a biosafety issue and also many other, technological issues. But I just, submit this idea to NSF, and I'm waiting for their, notice. So what will be your dream come true? Work with this electrogenic bacteria. Okay, so one of my long-term research goals is to develop standalone and self-sustainable biosensing system that can work independently and self-sustainably. Even in resource-limited areas, and I believe that the electrogenic bacteria, can provide the solution to pursue that goal. So that's my long-term research plan using this electrogenic bacteria. Could other organisms be used, like yeast or plant cells? As far as I know, yeah, there are some yeast, and the plant cells are has electrogenic capability, but I've not performed any experiments with this new species yet. Okay, so any other questions?
I don't think there are any other chat questions.
Right.
I have a question, and that might be way out there or not something that you're interested in at all, but is it possible to build something like a co-culture of bacteria that generate energy with some other strain, for example, a muscle cell that needs electrical impulses to control, for example, and electrical impulse activity? Would that be possible?
Yeah, good
at all?
idea. Yeah, definitely you can get a fund from NSF with this idea.
So good idea, but it didn't really work.
Yeah, good idea and crazy idea. I've never heard that. But yeah, why not? I think it's doable, theoretically doable, right? But I don't know. Honestly, I have no idea, Kate.
Yeah. Well, you didn't say it's absolutely stupid and would never work, so that's step one. All right. There is actually one more question in chat, I think.
Okay, are cable bacteria also electrogens? I don't quite understand. Cable bacteria? Is that bacteria with the nanowires? Oh, okay. So yes, many bacteria, bacteria strains can generate, can produce their own nanowires to transfer electrons from inside cell to the outside electrode. Some of the cables are reported to be conductive, while others are not. But there are several species that have conductive cables, like a Geobacter species. They have very conductive nanowires. They use this cables for the electron transfer to the outside electrode for high power generation. As far as I know, this Geobacter species is the best species for the power generation. Yes, the answer is yes. Okay, I don't think there's more questions.
Yeah, I don't see any more questions.
Right.
All right.
All right.
Thank
Kate.
you very much. Thanks
you,
so
Kate.
much, Tom. Thanks, everyone. And see you all next week. Bye.
Bye.